For 4 decades, neuroscience has compiled a precise, quantitative census of what Alzheimer's disease destroys. We can map the specific loss of pyramidal neurons in the entorhinal cortex, cholinergic projection neurons in the nucleus basalis, and the systematic decay within the locus coeruleus and the dorsal raphe. Yet one distinct population is entirely missing from this human autopsy count. There is currently no published, unbiased stereological estimate of dopaminergic neuron loss in the human ventral tegmental area. This gap in the data is a problem. The VTA is the biological source of the brain's mesolimbic and mesocortical dopamine. It broadcasts the chemical signals that determine if an experience becomes a long-term memory and how much effort a goal is worth. Experimental models have already mapped the timeline of this region. In transgenic mice, VTA dopaminergic neurons degenerate from 3 months of age. This structural loss occurs before amyloid plaques form and long before measurable cortical tau accumulates. Any accurate mechanical model of early Alzheimer's disease must account for how this specific dopamine neuron fails. Its silence directly initiates the cognitive and behavioral symptoms of the prodrome, years before the structural collapse of the cortex. This table maps dopaminergic vulnerability predictors: massive unmyelinated arbors, L-type calcium channels, and lacking calbindin. The substantia nigra fits perfectly, its massive arbor pushing it to the metabolic edge. Parkinson's disease obeys these rules, destroying the metabolically taxed substantia nigra while leaving the less taxed VTA alone. Alzheimer's models show the exact opposite. In the TG2576 mouse, we observe pre-plaque VTA cell death, while the substantia nigra remains intact. Even the classical markers are inverted. Surviving VTA neurons in these models express high levels of calbindin. This is not a baseline shield acting against the disease. It is a homeostatic upregulation, a defensive response triggered by an active biological stressor. Alzheimer's disease cleanly inverts the Parkinsonian vulnerability ranking. This indicates the operative insult is not a generic calcium or metabolic stressor, but a discrete pathogenic mechanism acting selectively on mesolimbic circuitry. Tracing that mechanism requires reconciling a direct contradiction in the experimental literature. One laboratory reports terminal somatic death of VTA neurons at 3 months. Another independent laboratory observes completely intact, yet hyperexcitable VTA neurons at 12 months. These studies describe different points on a single sequence. First, the SK channel regulatory brake fails, causing erratic firing. Second, the hyperactive cell's axon terminals thin and retract from their target fields. Finally, somatic death occurs. The cell body disappears last. This sequence creates a defined therapeutic window. Interventions like CK2 inhibition or levodopa successfully rescue synaptic deficits in early models, specifically because the cells are present but miscoding rather than physically missing. These conflicting studies are entirely compatible. Because cellular output and structural activity move in opposite directions, VTA failure begins in its corrupted code long before simple cell counts register physical death. Any hypothesis claiming early dopaminergic failure in Alzheimer's must answer a major clinical constraint. Dopamine transporter, or DAT, imaging is routinely normal in Alzheimer's patients. A reduced DAT tracer uptake scan is a confirmed diagnostic indicator for dementia with Lewy bodies. A normal scan serves as a differential marker, actively supporting an Alzheimer's diagnosis. This medical schematic shows the anatomical limitation of that scan. DAT-SPECT exclusively measures the nigrostriatal terminal field located in the dorsal striatum. The VTA's vulnerable projections are mesolimbic and mesocortical. Their terminals lie in the accumbent shell, the core, and the prefrontal cortex, areas the DAT scan does not read. A normal DAT scan does not refute VTA degeneration. It accurately bounds the lesion, proving the pathology remains isolated to the VTA targets and does not extend into the spared substantia nigra system. To understand the systemic cost of this targeted lesion, we look at the VTA's normal dopaminergic projection to the hippocampus. This circuit diagram of the CA1 subfield shows VTA dopaminergic terminals synapsing directly onto fast-spiking parvalbumin interneurons. The immediate fallout of VTA terminal loss is decreased D2 receptor activation on these interneurons. Without this drive, the interneuron degrades. NaV 1.1 sodium channels drop, perineuronal nets thin, and the failure of these fast-spiking interneurons removes vital inhibitory control, flooding the CA1 network with chaotic firing. This specific circuit collapse correlates cleanly with early Alzheimer phenotypes, presenting as degraded gamma oscillations and a strong predisposition to epileptiform spiking. Withdrawing this specific dopamine modulator does not silence the hippocampus. It forces the network into a state of chaotic disinhibition, providing a clear mechanistic explanation for early Alzheimer's hyperactivity. Dopamine also performs two distinct roles in memory consolidation. It acts as a 200 millisecond trigger initiating plasticity and a 12-hour delayed permissive signal determining if a memory trace will persist. However, mapping this function to the VTA introduces an anatomical complication. The locus coeruleus also supplies dopamine to hippocampal D1 and D5 receptors, and it also degenerates early in the Alzheimer's disease process. Current models suggest a strict division of labor between these two key nuclei, dictated entirely by their afferent triggers. The VTA response codes for absolute novelty. Rather than updating an existing context, its signal answers the fundamental question, have I ever seen this before? If VTA contribution is degraded, the predicted clinical phenotype is not global amnesia. Immediate encoding remains normal, but the patient experiences a total loss of novelty-induced memory enhancement. Without VTA dopamine, the brain retains its capacity to record an event. What it loses is the editor, the specific neurochemical signal granting permission for new experiences to be permanently stored. The systemic toll extends far beyond memory. The loss of VTA dopamine in the accumbens directly degrades the neural circuitry responsible for effort-based decision-making. This mesolimbic failure provides a biological mechanism for the two least explained non-cognitive features of the Alzheimer's prodrome: severe clinical apathy and pre-diagnostic weight loss. This ledger tracks the current evidence. On the left, confirmed mechanistic sequences in animal models, from CK2 hyperactivation to interneuron collapse. On the right, we have a glaring absence of human stereological cell counts. The animal data is thorough, but to convert this robust hypothesis into clinical fact, the field requires a direct, unbiased stereological count of the human VTA in Alzheimer's brains. Dopamine sets the biological price an organism will pay for a goal, and it grants the chemical permission for a memory to persist. If the VTA falls first, the Alzheimer's brain loses its will and its editor long before the physical hardware is crushed by plaques.